Hey everyone, in today's video we are going to discuss the cellular division process of meiosis, which produces gametes containing half the starting number of chromosomes. Now in our interest, this means going from a cell that contains 46 chromosomes to a cell containing half of that, or 23 chromosomes. Now humans, as we know, are diploid organisms, meaning that they contain two sets of chromosomes, and upon completion of meiosis, one diploid germ cell divides to produce 4 haploid cells, each containing 23 chromosomes. Just to clarify what these images mean, sometimes students struggle and they confuse some of the working components here. This here is just a single chromosome, and after completing S phase or going through S phase, you result in the sister chromatids here held together by their centromere. Before delving into the specifics of meiosis, I would just like to run through very briefly a general schematic of what is going on. So we start with one diploid germ cell containing 2 sets of chromosomes, and upon completion of the S phase, or DNA replication, you result in a cell looking like this, with these duplicated chromosomes being held together by their centromeres. Now, after undergoing meiosis 1, you result in the production of 2 haploid daughter cells, each containing one member of the homologous pair. Now, after undergoing meiosis 2, you're now going to result in the production of 4 haploid cells containing one set of chromosomes. Unlike mitosis, meiosis consists of 2 successive nuclear divisions, rightfully named meiosis 1 and meiosis 2. Now, before undergoing meiosis, the cell must duplicate its chromosomes during the S phase. After duplication is complete, the cell transitions into G2 and then enters meiosis. Now, looking at meiosis 1, the first stage of meiosis 1 is prophase 1, and during this stage, chromatin condenses, the nuclear envelope breaks down, and the homologous chromosome pairs begin to match up. Now, the transition between prophase 1 and metaphase 1 is something known as prometaphase, and during this stage, the homologs, or the pairs, they exchange genetic material, and the site where this occurs is called the chiasmata. After that happens, the cell then transitions into metaphase 1, which sees the alignment of the homologous chromosome pairs at the equatorial plate and the attachment of spindle fibers to those chromosomes. After the completion of metaphase 1, the cell then transitions into anaphase 1, and the highlight of anaphase is the separation of those homologous chromosome pairs to opposite poles. After that's complete, the cell then will transition into telophase 1, which sees the nuclear, or the reformation of the nuclear envelope around those newly migrated chromosomes. And then after that, cytokinesis happens, and you have the separation of the cytoplasms of those cells and the production of 2 haploid cells, and that is the end of meiosis 1. To save on time, I'm just going to talk through quickly meiosis 2, since it's very similar to meiosis 1. So after the completion of meiosis 1, the cell undergoes a very brief interphase with no S phase before entering prophase 2 of meiosis 2. Now, very similar to prophase 1 of meiosis 1, prophase 2 of meiosis 2 sees the condensement of chromosomes, the formation of the spindle apparatus, and the breakdown of the nuclear envelope. Moving on to metaphase 2, the chromosomes align at the equatorial plate, and then during anaphase 2, you have the separation of the sister chromatids towards the opposite ends of the poles, and then you have telophase 2 occur, which sees also, like telophase 1, the reformation of the nuclear envelope, and then cytokinesis follows, which results in the separation of the cytoplasms. Now, after this second division occurs, you have the production of 4 non-identical daughter cells that are non-identical to the starting parent, thanks to the crossing over that occurred between the homologous chromosomes during prophase 1 of meiosis 1, or prometaphase 1 of meiosis 1. So just recap, one diploid germ cell divides to give you 4 non-identical haploid cells.